All right, so we just finished talking about duplications, and next we're going to talk really briefly about deletions. But if you need a good table to study for some of the different deletions and duplications, their disorders, and then symptoms of those disorders, Table 8.1 really has a great breakdown of those different types of rearrangements that you can see in some of the syndromes that they result in. All right, so switching gears a little bit, let's talk about deletions. With duplications that we talked about in part B, we were talking about duplicating genes, and that resulted in an increase of gene material. Here we're actually talking about taking away genes or deleting certain genes out of chromosomes. So with those loss of segments within a chromosome, it can actually be big segments or little segments. Those deletions come in all sizes. So just an example of what a deletion would look like if we had, again, gene A, the centromere here in the center, and ending with gene G, and that was deleted to basically take off the whole back end of that chromosome where we were losing genes D, E, F, and G. You would have a really dramatic decrease in genomic material that would actually be available for transcription and translation. So, depending on how serious the deletion is, really determines how significant that coding or that region is going to be. So, if we have a gene that is going to be translating translating and or transcribing DNA material into messenger RNA and then mRNA into protein, then that is obviously a really important part of that chromosome. However, if you have a part of the chromosome that's deleted that is in between coding regions or in between genes, we call those introns. If you have deletions happening there, it's not as serious because it shortens the chromosome up, so there is an exact matching, but you're not deleting genomic material. So another deletion that can actually occur is if the centromere is involved. So deleting out an entire centromere can actually result in a lot of lethalities. So some of the things that happen when we get deletions of material is we get matching up of those chromosome materials except for those deleted genes. And what ends up happening is we can get some of those loops actually forming again to try to match up with its gene partner in in metaphase, and then we really have unequal separation in anaphase, so that the homologous chromosomes actually end up looking like this during pairing because they're trying to form that loop to get those genes to match up correctly. So some of the deletions that we see can really create imbalances in gene products. So especially in heterozygotes. So if we have two chromosomes that are homozygous, so they have the same deletions, it's really not an issue. Other than you won't have the protein product for the genes that were a part of that deletion issue. However, if you get heterozygotes, you really get a dramatic imbalance in the gene product that is going to be expressed at a phenotypic level. So some of those gene product imbalances are going to allow some recessive mutations that we see in the undeleted chromosome to be expressed because its partner is not there. So we call those expressions pseudo-dominance. So they're dominant because they're there, but normally if their partners on the other chromosome were there, they would not be expressed. In addition to that, some deletions are going to have some genes that have to function in pairs. So if one copy of the gene is not present and it's a gene that functions as part of a pair, then you're not going to get the correct upregulation or expression of that gene because it needs its doppelganger on the other chromosome. So what we end up with is haploinsufficiency. And we're going to look at another example of this here in just a few minutes. So again, just looking at what a deletion does, we've got material in the form of E, F, and G that are in question at this point in time. So what we should have here is E matching up with E, but we don't on this chromosome because it's been deleted out. So what ends up happening to match up the two G genes, we're going to need to form that hairpin loop so that we get that loop configuration, but we get matching of the genes against the deleted chromosome. So in an example of this situation would be notching in Drosophila or in flies. So notch is a series of X-linked wing mutations, and this is going to occur in flies that and those mutations really occur because portions of chromosomes have been deleted. So this notching phenotype is a dominant mutation. So you only need one copy of the gene to see it. If an individual is heterozygous for the notch gene, so it's possessing one mutated deletion and one normal genotype, then it will get a notched appearance like this here. So this is our normal wild type phenotype, and this is our notched phenotype. So the notched deletion is going to be designated as a capital N in a couple of these examples. This, we actually see an increased complexity to it because it's a deletion that occurs on the X chromosome, so on the sex chromosome. So keep in mind that Drosophila are going to be autosomal, so they have an XA ratio sex determination, and that'll become a little more apparent when we work through some of the other chromosomes and sex determination stuff. But for this example, what we end up getting is a homozygous female that would be lethal because they have two copies of that notched gene. So if those notched genes occur together on the X chromosome, that individual is not going to make it to birth. So those larvae die. Another lethality is in males. We can actually see a hemizygous lethality where just there's only one X chromosome that's possible in a male, and if it has that notched gene on it, then it's going to be a lethality. Some other conditions in humans that we see this type of deletion rearrangement occur in is Creutzfeldt syndrome, which is actually French for cry of the cat. And this syndrome, if it occurs in a child that is heterozygous, then that individual tends to have a very small head, wide eyes, a round face, and is intellectually disabled. So those individuals are going to have deletions on chromosome 5, specifically on the short arm of chromosome 5. A second syndrome that we see happening in humans is Wolff-Hirschhorn syndrome, and this is going to be where we actually see a deletion of a tiny segment of chromosome 7, and that's going to cause the gene elastin to not be expressed, and that's going to really cause distinctive facial features in those individuals and they're also going to have heart defects, high blood pressure, and some cognitive impairments that probably most likely in the form of some sort of retardation. So these deletions can have major issues from a phenotypic standpoint. So again, spend some time studying Table 8.1 and get a good handle on some of the duplications that can occur with rearrangements, as well as some of the deletions and some of the syndromes that those result in.